acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past, present and emerging, and embrace their continued connection to this place. I'd also like to extend this respect to elders of communities from where people might be joining remotely. It's a real pleasure to introduce today's speaker, Ramyar Molania. Ramyar joined my lab in 2018 after completing a postdoc with Alex Dobrovic from the Olivia Newton-John Cancer Centre and Terry Speed. And his PhD was focused on normalization problems. Since joining my lab, he's worked on integrating large-scale RNA-seq datasets from cancer, as well as single-cell RNA-seq and spatial data. You're probably all aware that we have a crisis of reproducibility in biomedical research, and today you're going to hear about a particular aspect of that crisis, the crisis of normalization, possibly a crisis that's underappreciated. Ramyar is going to talk about work that really started during his PhD with Alex and Terry, and has continued since joining my lab. Ramyar.
All right, thanks everyone for coming and people who are listening to me online. Today I'm going to be talking about a pretty familiar topic, which is crisis in irreproducibility in biomedical research. And I'm going to highlight how important the study design and data normalization are in mitigating this effect crisis. Okay, over the last decade, a massive amount of data has been generated. Thousands normal and cancer tissues are profiled at the different molecular modalities in a large-scale project like a Tcga or GTEx. In the CCLA, we have an expression profile of 1,000 human cancer cell lines, and recently in a human cell atlas, all the individual human cells is going to be profiled at the single cell level. One of the major repositories of gene expression data is the GEO, and there are over 2 million samples in that repository. And GEO doubles in size every 2 years on average. However, irreproducible biomedical research costs $28 billion a year. That stat is just for the United States. It would be higher, significantly higher over across the world. And I should say that funders, researchers, journals, institutions are all involved in this crisis, and they must do their part for reproducibility. And I'm going to say that that is a perceived crisis, and it shouldn't be seen as a bad news, but as an opportunity for major improvements. There are several key reasons have been discussed in the literature and by people for irreproducibility in biomedical research. I've listed a couple of them here. Definitely poor experimental design and the use of inappropriate methods for data analysis, including data normalization. Insufficient methodological details in the literature. Insufficient technical skills. The slow execution of the experiments. Sometimes there are deviations from the protocols, and lack of rewards for reproducibility and this incentive for irreproducibility. There are papers still that we know that the results are not correct, but they are getting citation and citation every day. There is no doubt that good experimental design and appropriate data analysis methods decrease false discoveries compared to the poor experimental designs. But the point here is, even if you have a good experimental designs, if you use the bad normalization method or inappropriate methods, still you end up with a high false discovery. And pretty the same when you have a poor experimental design, and even using appropriate methods, you're going to end up with a pretty high false discoveries. Yeah, you may say that good experimental design costs more compared to the poor experimental design. Whenever that I suggest my collaborators to have a technical replicates, they say it's going to cost more. But the point is here, the data analysis is going to is going to cost less for the good experimental design compared to the badly designed experiments. And at the end of the day, we may save budget if you have a good experimental design. All right, what is the characteristics of the poor experimental designs? Raw replicate samples. If you have one sample versus one sample or something like that, you're gonna have insufficient statistical power. No or poor designed technical replication across different batches. If you have a different batches, like different reagents, different platforms, if you don't have a technical replicate, it's going to be very hard to estimate the batch differences. And no poor negative and positive control genes. In this talk, negative control genes are those genes that are roughly constant across experiment, like housekeeping genes. And the positive controls are those genes that we know the biology. And we're going to check that after normalization. And the major one is a confounding, when the biological factors of interest are largely confounded with the batches. I'm going to have a cartoon here to explain it. What does it mean? Let's say you have got 10 samples with treatment A, whatever it is, and another 10 samples with treatment B. And you're going to use the same platforms, but using two different reagents, Z and Y. And if you put all the one treatment in one batch, another treatment in another batch, it would be very, very difficult to distinguish the batch differences from the biological differences. That is really big problems. And it still is happening in these days. I said normalization. What does it mean in the gene expression world? In the normalization, we adjust for or removing of any variation that are unrelated to biological factors of interest. And those variations can compromise downstream analysis. Collectively, we call this variation unwanted variation. And there are different sources of them in the gene expression dataset. The first one is a varying level of RNA input. If you add more RNA, you get more signals or sequencing reads. We need to adjust for it. Gene lengths in the sequencing-based assay. And the longer gene, the more reads. We should adjust for that one as well. There's a paper published recently that sample-specific gene length bias can cause problems in RNA-seq data analysis. You've got the GC content, and the popular one is batch effects. If you are using different reagents, different protocols, different platforms, even date can cause batch effects. I'm going to highlight this point here, that each source of unwanted variation may have different effects on the data set. If you have a library size, we should adjust for library size, and for batch effects, for GC links, they have different effects. It's not just one effect. They don't have a one effect across all the data set. And if you wonder how many normalization we have plenty of normalization out there for gene expression, and maybe around 100 even for single cell. And sometimes they don't deliver the same results. I've listed a couple of them here, but regardless what kind of normalization we are using, we always need to look for evidence that normalization works. It helps and doesn't hurt, and because sometimes we've got that the normalization can hurt the data compared to the raw counts. In this talk, in our analysis, we have 3 levels of evidence to show that our normalization works. We have a weak evidence, starting with a statistical plot, like a PCA, RLE, and other tools. Intermediate evidence, we have a technical replicates, because at the same samples repeated twice, the similarity of technical replicates should be checked after normalization. And then strong evidence is if the normalization can reveal known biology or positive controls, that's a strong evidence that our normalization works. I'm going to explain quickly 2 plots that I'm going to show them a lot in this talk. One is relative log expression plots. In the gene expression world, usually we get the gene expression matrix like this. We have got genes in row and samples in columns, and we get signals or count. And then we kind of transform this data set. For individual gene, we take a median, and we subtract the medians of that gene across all samples, and we get a relative log expression. And then for each samples, we do box plots. When there is no any unwanted variation in the data sets, we get a good RLE plots, and it means that the medians are centered around 0, and boxes are are pretty similar to each other. That shows that you don't have an unwanted variation. But what happens if you have an unwanted variation in your gene expression data set? The median is going to deviate from the 0, and boxes is not going to be similar to each other. We call these plots bad RLE plots. Another plot that I'm going to explain, I'm going to show you this plot a lot today, is the principal components, and is a kind of cluster of samples based on the similarity. And for example, if you have 6 samples here, these 3 is from the one biological condition, another 3 is from another biological condition, and let's say sample 1, 4, 6 are profiled in the laboratory X, and these 3 samples are profiled in laboratory Y. And when we do a principal component, we get these kind of things. We need to color the principal components. For example, if you color that this PCA, you can see that the biology is separated in a PC1, there are 2 clusters, and batches are separated in a PC2, which is the way that we kind of look at the principal components. Okay, the outline of my talk. I'm going to share with you a 6 years experience, or 6 years story in terms of the normalizing data set. And it was, we started from a nanosecond gene expression data, and the plan was to go to the Tcga, which is the largest project in cancer, to validate some of the nanosecond. Pretty reasonable approach. People do it a lot these days. That was supposed to take a couple of months because everything should be straightforward, but it took 6 years because when you're working on a badly designed experiment, everything's going to be complicated. And in the first part of my talk, I'm going to talk about nanostring, introduce the platforms, talking about the nanostring data normalization, introduce the REV tree, which we have developed in the Terry's lab, and I'm going to show you how we use REV tree for removing normalization, unwanted variation from gene expression data from nanostring. And we published a paper 2 years ago, and if you are interested in more details, you can read that. And in the second part, I'm going to talk about the Tcga, sources of problems there, and I'm going to talk about a new approach that we call it pseudo-replicates of pseudo-samples. And as a paper of this is going to be available online pretty soon. Okay, nanostring is a pretty good platform. It's developed to directly detect any type of nucleic acid. It's a highly multiplex assay. You can profile up to 800 genes. It's a probe-based system, and for data analysis, there's a software called Ensolver. And in terms of the experimental pipelines, it's pretty straightforward. From the sample preparation to get the count, it takes 1 or 2 days. And the nanostring claims, 2015 to 2019, when we were working on the nanostring, they were saying that there is no need for any technical replicates because the platform is robust. And there is no need to use any advanced normalization. The Ensolver is fancy enough. And in 2020 to present, because when we published our paper, they said the technical replicates are required, and user can use any normalization that they want. This is a good improvement for them. Okay, I'm going to briefly talking about our nanostring data set. We have got the 108 normal and lung adenocarcinoma tissue samples. We profiled 600 genes involved in DNA repair mechanism, and samples were profiled across 15 nanostring cartridges. These are the cartridges, number of samples, and this is a date here. In terms of the tissue distribution, is not the ideal design. The reason is you can see here. This color is all normal. And this is all cancer samples. You can see in around 15, which is 1 year apart from the latest, around 14, the majority of normal samples are here. This is not a good design. I would like to have all the normals across the batches. But fortunately, we had technical replicates. Each 2 points or 3 points connected with one line is one technical replicates. For example, this sample is repeated here. This sample is repeated here. And I would say it's not ideal. It wouldn't be ideal as well because I would like to have replicate in round 13, not having a lot of replicates in round 5. No, we don't have a replicate here. I would like to have 1 here as well to capture the variation of round 13 as well. All right, we applied the nanostring the way that nanostring recommended, and it failed to removing unwanted variation. I'm going to show you the RLE plots Each box is one sample. Each color represents one batch. You can see the variation. Of course, is unnormalized data set. And on the nanostring data set, it improves, but is not a good RLE plot. You can see there is problems between batches. The boxes are not similar to each other. It shows that data is not well normalized. And how about the technical replicates? Because as I said, the similarity between them should be evidence. This is one pair of replicates, and each point is a one gene, and all genes should be in this identity line, because this is a technical replicate. The same samples repeat twice. For example, one gene here, you can see there's a huge expression in one replicant, and when it's repeated, there is no expression. It's a huge differences. How about the revealing known biology? At that time, there was, we were interested in the correlation between these two genes, and there was plenty of references that showing in the protein expression level, there's a positive correlation between these two genes. We look at the macro array from this study, and we kind of see that correlation. And when we look at the nanostring, there is like a negative correlation. It's inconsistent with what we have observed or seen in the literature and in this data set. We were pretty unhappy with this kind of normalization, and then I decided to use a method developed by Terry Speed and his colleagues. It correlates removing unwanted variation tree, is a way of normalizing omics data, and it makes essential use of negative control genes that are roughly constant across all samples, like housekeeping genes, and use technical replicates. Any differences between technical replicates will be used to estimate unwanted variation from the dataset. In situations when technical replicates are not available or technical replicates are badly designed, if you put all technical replicates in one batches, they are not going to be useful. It should be distributed across all batches. In this situation, we propose a new approach. We call it pseudo replicates of pseudo samples. And this is the RUV3 normalized data set. You can see the medians are set around 0, and boxes are pretty similar to each other. It's a big improvement compared to nanostring normalization. In terms of the intermediate evidence, you can see that the technical replicates are pretty similar to each other in the new normalization. And in terms of the known biology that we were looking for, you can see that correlation we observe in the RUV3 nanostring that we couldn't see in a nanostring normalization. At that time, we reached out to the nanostring and said, okay, your method should be improved because we couldn't get the satisfactory normalization. And they came to here and they saw all the results, but they didn't listen because they said that our method is sexy and we don't need any fancy normalization. That's why we decided to look at the publicly available data set to see how the nanostring normalization worked for them. The first one is a paper published in Science. There are 2,000 samples, and we showed in my paper that their normalization failed, and unfortunately they reported a lot of false discoveries. Another paper that we look at it is another paper in Science. There are 2,500 samples, the same problems happening here. Another one is here, published in GCI Insight, and again, the nanostring normalization failed and led to a lot of false discovery. All the papers are intensively discussed in my paper that we published. If you are interested to know more, you can read my paper. I'm going to take you very quickly through this example here, published by Plequin in 2016. And this is a pretty big paragraph. You don't need to read the sentences. But this is the way that they normalize the data set. It's pretty kind of like they explain everything. But the problem is they haven't shown anything to show the performance of the normalization. Even in the supplementary file, even one PCA, one RLE plots. They mention that there's a problem, we have a batches, but they haven't shown anything. Okay, I'm going to show you how they designed the study. There are 3 tissues, like colon, rectum, and terminal ileum. The disease status, we have different disease status. They profile up to 700 genes. There are 15 housekeeping genes for normalization, and there are around 1,000 samples. And they use 3 nanosecond reagents across 3 years. They change reagent, they change kit. That's alarming. Okay, you have to use different reagents. And this is the tissue distribution, and you can see this at batch 2, 3, 4. If you wonder what's happening for batch 1, this is my question as well, because the way that they annotated the sample is batch 2, 3, and 4. And in terms of the tissue distribution, it's pretty good design. You can see all the tissues and the disease status are pretty uniformly distributed across all batches, which is good. And they have technical replicates, which is great. You can see each point, one sample repeated here. This is within the batch technical replicates, one sample here, repeated here across the batches. That's great design. And I'm going to show you the Plequin normalized dataset, the data that they reported in the paper. These are the principal components colored by batches. Each point is a sample. As you can see here, that the samples that are coming from the batch 3 are separated in PC2. There's a batch effects left in the normalized dataset. And when I color, because I said all the tissues tissue are across all batches, and we don't have a confounding here. You can see that batch has got all tissue, all the colon, rectum, ileum. This is clearly batch effects left in the normalized dataset. And when you look at the RLE plots, because there's 1,000 box plots, I'm just highlighting the medians. There's a medians, and I'm showing you the interquartile range. You can see that this is a bad RLE plot. Medians are not centered around 0, and boxes are very different. The question is here, what is the consequence of these batch effects in the downstream analysis? I'm going to show you one of the examples. We have plenty of examples in my paper. This is the expression of CCDC 101 in the y-axis across all samples. Each point is the one sample. You can see the expression of these genes goes high, 64 times higher in the batch 3 compared to the other batches. And when we look at the expression of this gene across these disease status, this gene has been reported as a DE gene, as a differentially expressed. This is the mean here for each group. And you can see there's a shift in the mean, and that shift is because of these samples, because of batches. And that's why they call it DE genes. That has a consequence of the differential expression analysis, and has got a lot of other. We have got a lot of examples for this study here. Okay, I'm going to mention here, because it's a well-designed experiment, and we have an appropriate method, it took me literally 2 days to get that data normalized, where in those science papers took me 1 year, because the design was pretty bad. It was a pretty complex study as well. And that was the Plequin normalized dataset. We get the REV3 normalized, which is center medians around 0, boxes are pretty similar. That was the Plequin, the PC of Plequin normalized dataset, and here's the PC of REV3. You can see the batches are mixed. You can't see any batches anymore, but there's 2 clusters here. That cluster is the tissue differences that we expect to see in this dataset. This is the ileum, and this is the colon and rectum. If you wonder why we use REV3, why not other normalization method, we have done. I'm going to show you in a minute, but show you the genes that were DE. That was the Plequin, and we get that expression pretty right in the REV3 normalized dataset. And this gene is not DE anymore. And we look at the macroarray dataset, which has got the same disease status, and this gene is not DE in that macroarray dataset. We could use it as an independent dataset to confirm this result. I should say that when you look at the row counts of this gene, it's similar to this one here. That is the big artifacts of the bad normalization method. At that, even if they use the row counts, they could get better results. And we use other methods, like a CPM is a count per million. There's a batches, there's a PC1, PC2, 1 against 3, and 2 against 3 here. There's a batch effects. It didn't work. We did upper quartile normalization. They are pretty popular methods. Still, batches are clear. In the quantile normalization, in the combat. And this method, combat, has been designed for such problems. we can't see batches in the PC1, PC2, but we can't see biology anymore as well. It removes everything. It's not removes batches plus biology, and that's why I decided to use REV3 at that time. A summary here. None of those things are good platforms. Especially, it works great for FFP samples, but the data analysis should be improved. Positive and negative control genes, because it's not the whole genome, it's not the whole transcripts. You need to have a positive and negative controls. It's 100 or 800 genes. Having the suitable positive and negative control genes, technical replicates are critical for designing non-nesting assay. And as you saw, the selection of normalization can greatly influence downstream analysis. And I think, and what we have been seeing, that REV3 is really good method for normalizing non-nesting dataset. All right, as I mentioned, we were planning to look at the non-nesting and go to the Tcga to confirm it. That was a plan of my first PhD project. And I'm going to introduce Tcga and show you what's happening in that project. Just very briefly, the project was launched in 2005 and ended in 2018. The Tcga research network by that time collected more than 11,000 samples, which is great, over the 33 cancer types. And for individual patient, we have different molecular modalities like a mutation, copy number, gene expression. And the data have been used for research and clinical applications, and it has been estimated that there are more than 10,000 publications that use the Tcga. And there are tools, I think it's pretty popular for biologists, like a CBio portal, Tcga Firefox, and you can go to this website and without downloading the data and coding, explore the Tcga normalized dataset for mutation, for expression. In terms of the collecting and processing samples, the samples were collected across 831 laboratories and then shipped to 31 genomic centers for profiling. Within each center, samples were allocated to different plates, the sequencing plates. It's a pretty complex study. For example, here in the Tcga colon adenocarcinoma, there are 479 samples. 20 laboratories were involved in providing tissue, 24 plates, and data were generated across 4 years. But I'm going to take you to the breast cancer RNA-seq today. We have done colon and rectum, but I'm going to show you the breast cancer. There are 1,100 samples. There are 40 laboratories, 39 plates, and data were generated across 5 years. For people who are familiar with the breast cancer, we have PAM50 subtypes, which is HER2, basal, luminal A, luminal B, and normal leg. These are expression subtypes. We can use it as a positive control to see whether we can see it in our clusterings or not. And how the Tcga normalized the data set, they use FPKM, which stands for fragments per kilobase of transcript per million mapped reads. It adjusts for gene length and library size, and then they use the upper quartile normalization to remove some other problems. And there are also macroarray gene expression data of 500 of these samples. We can use these macroarray as a kind of independent data set to confirm the RNA-seq results. Okay, I'm going to show you the PCA plots of the Tcga normalized data set, that one that are frequently used in the papers. And colored based on the PAM50, you can see basal is here, and there's a HER2, luminal A, luminal B, and normal like here. There's a PC1 against 2, and the other two, PC2 and PC3. I'm going to color the same PC by time. If you look at the PC1 against PC3, you can see the yellow color, which is samples profiled after 2012. They are separated in PC3. There's some variation. What's happening for samples after 2012? We thought that is a time effects that is in a data set, but we emailed Tcga and said what is happening. And they say we use two flow cell chemistries. They changed the reagent after 2012 to profile the rest of the samples. which is fine. That's fine because it's a large project. They can change the reagent. But the question is here, why that information is not available? Why we should contact you and say what's happening? Why you don't make that? You haven't made that details available. That is a flow cell chemistry that is left in the normalized data set. But if you look at the basal here, for example, and look at it from the principal component to here, there's a pretty big variation across basal. What causing this spread for even within each subtype? When we color the same PCE usually based on the purity, you can see here this is a low pure samples, and this is a high pure samples. Tumor purity is the percentage of purity, the percentage of the cancer cells in the tissue, because we have got the cancer cells plus stroma and immune cells. We call it purity. The 1, it means pure cancer cells, and 0 is pure immuno or stroma cells. And you can see the PC2 is a purity. And you may say, okay, purity is a biological factor, but people are talking about that, and they're connected to the survival or gene expression. I'm going to show you whether is it wanted variation or is it unwanted variation. All right. I mentioned CBio portal. And when you go to the CBio portal and look at the Tcga normalized data set, and look at these 2 genes I selected randomly from my results, there's a negative correlation between these 2 genes. This is a CBio portal. I just took a screenshot. There's an obvious negative correlation between these 2 genes. The question is here, is it real? Should we go to the experiment, to the lab and assess it, or write a grant or something like that? I'm going to show you what's happening. I'm showing you here a heat map of 140 genes that are highly affected by flow cell chemistry. These are samples here and genes here. It's the expression. And this is one set of flow cell chemistry. This is another set. And you can see the expression of these these genes here are pretty high and get lower in another set of flow cell chemistry. And this expression of these genes are pretty high and pretty low and get high here. The point here, when batch effects occurs, a subset of genes are affected in different ways. That's why these things make normalization much complicated. Anyway, to quantify this, and there are another 2 patterns here, one pattern here, another one here. It's like a within batch problems. It's like 2 other batches here. To quantify these complex batch effects, we take these genes here and we score samples using sample scoring to see how enriched these genes are in the samples. You can see here, this is a scoring that we get from across samples. We call it batch score. You can see there's a flow cell chemistry here, and we can see these 2 bump here in that score. And we divide the samples based on the score into 4 groups or 4 sub-batches. We call it like 1 batch here, another batch here, another batch here, another batch here. Let's go back to do the genes between the correlation between MAP3K2 and CERT6 here. If you look at the correlation of MAP3K2, there's a correlation here with the batch score. There's a negative correlation with that. And you can see how these complex batches make that correlation with that MAP3K2. And for the CERT6, there's a positive correlation. And when you plot these 2 genes against each other, they have a negative correlation because of those complex batches. And when you look at the Tcga breast cancer macroarray, it's the same samples, only 500, but using macroarray, we can't see that correlation. That is a result. That is a kind of result of the unadjusted flow cell chemistry in the Tcga. I'm going to show you another gene here, 2 genes here. It seems there is no correlation. And the question is are they really uncorrelated, these 2 genes? And showing the 2 genes color based on the PAM50, there is no any PAM50 effects on these genes. I'm going to look at the correlation of these 2 genes within each batch, those batches that I explained in the heat map. There's a 1 batch here, another batch here, another batch here, another batch here. And this block dashed line here is overall correlation. Overall, we don't have a correlation overall, but within each batch, we've got a pretty good correlation. And when you look at the Tcga breast cancer macroarray, we get that correlation. And this correlation between these genes has been missed because of the batch effects. I'm going to show you another two genes, ZEP2 and ETS1. And there's a positive correlation. You may say, oh, this is not the example of the batch effects. Maybe because of batch effect, these two genes are highly correlated. I'm going to show you the expression of two genes based on the, and color based on the batches. Is that the batch effects? Is it, maybe it's real because it's not affected by batches. I mentioned that each source of unwanted variation have their own effects. When I look at the expression of ZEP2 with the purity, there's a negative correlation. There's high pure samples, low pure samples. For that gene, there's a negative correlation as well. When these two genes have a negative correlation purity, they can have a positive correlation to each other. You may say, okay, purity is biology. But we can prove it. We can prove this correlation we can use a LCM dataset, is a laser microdissection. In that dataset, they just capture the cancer cells. And they have expression of only cancer cells, not a stroma. And we look at expression of this gene is not that highly correlated. And this correlation is a result of variation that introduced by purity. And you don't need to look at each plot. I'm just showing you very quickly the breast cancer, but we look at all the Tcga, or 11,000 samples, and the larvae cells that I haven't said anything. Purity and batch effects are the major problems across all the Tcga dataset. How we can normalize them? We use different methods, like a CPM, all the methods. All of them didn't deliver satisfactory normalization. We wanted to use REV3. But the problem is here that we don't have a technical replicates in the Tcga. It's a 500 million projects. They haven't spent anything for technical replicates. Then they knew that it's going to change the chemistry, but I don't know why they don't have a replicates. The way that we're going to deal with this problem is a new approach that we have been developing. It's called pseudo-replicates of pseudo-samples. I'm going to explain it briefly here. Imagine that you have got 3 batches. Within each batch, you have got the 3 biological factors, like let's say PAM50 subtypes for breast cancer, for example, or whatever that you know is a known biology for your data set. And then for each group of biology, we take a gene expression average, and we create one pseudo samples by averaging expression. And we do it for another batch. We do it another batch. And these 3 pseudo samples are a set of pseudo replicates for that example here. And the differences between them are mainly unwanted and the batch effects here. And we did it for another groups here, for other biological groups. We get another set of replicates, and we did it here as well. With this approach, we were able to remove a lot of problems from the Tcga. And you may say, okay, I don't know everything about my data set. I know one subtypes, but I don't know other things. But as long as you have got one known biology shared across batches, you should be able to use the pseudo-replicate of pseudo-samples approach. But if you have an experiment like that you have all biology in one batch, another biology in another batch, we most likely not be able to help you even with that example. Because you can't get any pseudo-replicates across this example here. All right, to show you the REVT normalized data set, that was the Tcga normalized flow chemistry problems, and we mixed it here in the REVT normalized data set. There is no, we reduced that effects in the REV3. In terms of the PAM50, we haven't removed the PAM50 signals. We have the basal, we have got the HER2 here, luminal A, luminal B, a bit of normal like here. And we have done a lot of statistical tests on this analysis, and all show that these clustering are better than Tcga. And that gene, these 2 genes that has a negative correlation because of complex batches, we can't see it in REV3 anymore because we removed the batch effects. And as I mentioned before, there are plenty of these examples, not only in Tcga breast cancer, in all the Tcga RNA-seq dataset. And for that one, the correlation between these 2 has been missed because of the problems. We saw it in a Tcga breast cancer macroarray, and we get it pretty right in the REVT normalized dataset. And for the purity one, because I haven't shown you how to remove purity, because for the sake of time, but we were able to remove purity as well from the dataset. And as I said, that the LCM data showed us there is no correlation, and we don't see that correlation in RUV3 because we reduce the variation caused by purity in the RUV3 normalized dataset. What other things are happening? There are plenty of downstream analysis are affected by batches, purity, large size effects in a Tcga. One of them is survival analysis. There's a gene that, sorry, I forgot to put a reference here. There's in the literature, they showed that the expression of a STAB1 gene tumor grows in the breast cancer. When you look at the Tcga, the expression of this gene is not associated with survival. And when you look at the RUV3, and just so you can see that the gene is associated with the survival. And the low expression is a better prognostic, is consistent with what we have seen in the literature. And again, there are plenty of genes that you can't see the survival in the Tcga normalized data, and we could see it in RUV3. Just the summary of the Tcga. Tcga is a great resource. Is the largest project for cancer research, but if we need a better normalization for the data. Their normalization is okay, but it doesn't deliver a very satisfactory normalization. Removing unwanted variation is an essential aspect of analysis of large genomics dataset, any genomics dataset. Library size, tumor purity, batch effects are major sources of unwanted variation across all Tcga RNA-seq dataset and can compromise several downstream analysis. RUV3 with the pseudo-replicates of pseudo-samples is valuable for normalizing Tcga. And maybe removing those problems from Tcga may yield new insight into dataset. Okay, the concluding remarks. Reproducibility is a perceived crisis, but as I said, it shouldn't be seen as a bad news. We should just improve it. It should be an opportunity for the major improvement for reproducibility. As I said, research funders, journals, researchers, and institutions must do their parts for reproducibility as well. Well-designed experiment, suitable normalization methods are essential to drive meaningful biological results. And the positive, negative control genes, technical replicates are critical in designing gene expression experiment. And the effectiveness of any normalization method should be assessed, regardless what kind of normalization you are using. You should have evidence, at least for yourself, if you don't want to publish it in your paper. You should have those evidence that your normalization works. And as I said, RUV3 with the pseudo-replicate of pseudo-samples approach opens up a great opportunity to normalize a large and complex gene expression dataset. Even I recommend to have a technical replicates, but if you don't have technical replicates, still that approach may help. Yeah, I would like to thank all people who have been helping me in these two projects, specifically Terry and Tony, for their countless great ideas, discussion. We had over 100 meetings for Tcga. I think they are sick of me. And then thanks to Luke for all the great discussion about library size and his feedbacks about my talk. Gabriel for discussing things about pseudo-replicate and those things in the RUV3. I would like to thank Alex Dobrovic. He's my main PhD supervisor. All these things started from my PhD. And all other people that have been helping me in this project. I would like to finish my talk with this quote from Fisher, and I'm happy to take any question.
We can start with questions from the audience. Annie had your hand up first, I think.
So that was really interesting. So what the logical consequence of this is for you to start a website where you let others examine the Tcga data set with your normalization, which should be more beneficial for them.
Yeah, that is a plan that we're gonna hopefully to have a website that you can have not only for TCGA, even for your own experiment that you can upload, as long as you know some subtypes about your data set, because we know the biology a bit to be able to.
I understand that would also be good, but TCGA is used by many people a lot of times, and if it's that bad, then it will lead to a lot of false results if they go on using it in the way that's been normalized. So I think it's important to put up a TCGA corrected.
Yeah, the approach that we have been doing is to let people know first and then publish our paper. We let people that all that paper from the authors from the science or then the paper that, okay, you have got the mistake, you have done something wrong in your analysis, but they didn't want to listen. We publish the data and then they listen. We let the TCGA know that, okay, there's problems, even for the flow cytochemistry in the breast cancer, and they said it's not a major variation. And you can see that there's a lot of problems. We're going to publish our paper in a hope that they kind of use the new normalized data set or they change the normalizations. Hope something happens.
Matt has his hand up.
So the data set, analysis that you did on the RNA-seq from the TCGA, that was on FPKM adjusted?
Was FPKM plus upper quartile, yeah.
Yeah.
Just
No,
when you
Oh,
finish
yeah.
your question, you
Oh, sorry.
question.
I thought, yeah, okay. Said your question, I'm just, okay.
Yeah, so your analysis was on FPKM
Mm-hmm.
counts as well.
Yep.
Would you like to comment on availability of appropriate data for being able to do this sort of reanalysis and how challenging that is with these studies as well? Like raw counts, for instance, rather than FPKM counts.
The question is that I've been using the TCGA FPKM plus upper quartile normalized dataset, which is the most current version. And the question was, how about the new normalized or some people that did normalization?
Well, raw counts.
Yeah.
Raw data. Yeah.
Misunderstanding. He analysed the raw counts.
So he analysed the raw
He
counts.
compared
that
it
was
to
really
FPKM.
So yeah, okay. So the RUD.
Repeat that.
Okay, repeat this one. Yeah, we reanalyzed the raw counts, even for the nanostring and the TCGA, because our method requires raw counts. And there are some other normalized data set by TCGA, by other people that use the TCGA, like a CPM, TPM, but all of them have problems.
Rory's had his hand up next, and then you're next.
Right, so I was particularly excited when you showed the purity stuff, because of course that's something which exists in the samples, but hopefully you could remove the effect because you understand it. And I was slightly disappointed you couldn't get rid of all of the purity effect, and I wondered whether it was because you'd considered purity to be an absolute measure and you weren't willing to just fit your own measures of purity. But you should be able to infer the purity from the samples.
The question was, we were not able to remove all the purity signals, and because the data is complex, and that the pseudo and we try to reduce the variation. We may be able to remove all of them, but we reduce it a lot, and the new results for the breast cancer, when we remove purity, is very, very consistent with the LCM expression. And there are some tiny differences. We show that we haven't removed all of them, but we can do it. But that is just major improvement. I'm not going to say this is the final TCGA normalized data set. I'm going to show you the way that you can get the final expression or that remove everything.
I've got some Slido questions in a second. There's a few more hands up the back.
I always find it a little bit scary when I see kind of lots of variation get squashed down into a straight line by a normalization method. And I was just wondering if you could comment on how much sort of biological or real biological variation would be lost in the process. So and I assume that these would be kind of relatively subtle, but nevertheless real differences. Can you have an estimate of how much is lost as well as kind of how much is gained by the normalization process?
Yeah, the question is how much biology we may remove in the normalization process. It's all about improving. It's about the major improving the expression. And as I said, we have a lot of evidence. I showed you a very, very quick, or a tiny part of it, like a very large project. And we have done a lot of analysis as long as we can, as long as we know about the data set that we haven't removed biology. Maybe you say, okay, oh, you remove that signal here. That's good. That is another known biology that can help us to redo the analysis and keep that one as well. The more biology we know, the better job we can, because the design is badly designed. I can't show you why TCG is badly designed. They could even, without technical replicates, they could even do a better job. But for that kind of complex and badly designed experiment, we improve it a lot as long as we know, as long as we check, as long as our assessment tools show the effectiveness of the normalization.
I'm going to read a Slido question next from Anonymous. Do we see similar unwanted variation in DNA data sets, for example, whole genome sequencing, etc.? Is tumor purity still a big effect in DNA-based data sets?
There is a paper published 2018. They showed there is a big batch effects on Tcga DNA based assay. It has been published for DNA.
Marie-Liesse.
Beautiful talk. I find it interesting that you use microarray data to use as your comparison and validate your normalization. Because to me, RNA-seq was a big improvement from microarray data. So I just find that interesting. And I guess I wanted to know, because purity will be an issue on microarray data.
Mm-hmm.
So when you analyze those microarray data, how were they normalized? And is purity taken into account? Have you looked at the purity effect and the batch effect, all of that in the microarray data?
Yep. The question is, I've been using the macroarray gene expression datasets, whether from Tcga or publicly available datasets, to kind of confirm what we have seen in the Tcga RNA-seq dataset, where RNA-seq is better than macroarray or has a big improvement, something like that. For the batch effects, whenever that I want to use the publicly available dataset, I assess the normalization as well. It's not like, okay, that is normalized, because I don't trust any normalization unless I assess the expression. When the data is well normalized, I use it for the batch effects, and we have evidence. There's a batches, and it's been removed. For purity, that's why I couldn't use the Tcga breast cancer macroarray. It's got the same purity issue. I used the LCM dataset. It's a laser capture microdissection. In that, which is a 500 breast cancer, LCM breast cancer samples, there is no purity there. Even for that example, I ran the purity estimates, and I saw there is no purity in the dataset. I had another LCM. When I ran the purity estimates, I saw the purity even in the LCM dataset. I used that one that I'm pretty confident there is no purity, and then compare the result to the RUBT normalization. Yeah, that's why I couldn't use the Tcga breast cancer macroarray. It has got the same problems. But for the batches, we have evidence there's a batches. We can't see it in the macroarray, because macroarray is much clearer in terms of the batches in the Tcga.
Just before we have our next question from the room, I know YouTube broke at the start of the talk. The talk was so good. And so that some of our viewers, our remote viewers, are probably lagging behind. They're able to start rewatching it, but about 10 minutes late. So for those viewers, feel free to email Ramyar your questions. More questions from the room? Otherwise, I'll read another one from Slido. Interesting talk from Anonymous. Can we believe anything published about Tcga RNA-seq?
Yeah. As long as you because people are not interested in the whole biology of the Tcga. If you are interested, for example, in this problem, and you can capture it and you know that is all right, you have a validation, whether in experiment or other things, yeah, that's fine, just go ahead. But if you want to do discoveries, If you see the correlation between two genes, oh, that's great. I'm going to go to the experiment to check it, and I'm going to write an NHMR sequence, something like that. You need to look at the other data sets to make sure that is the right.
Can the normal users see all this information on the Tcga?
Yeah, that's why I had a screen. Okay, sorry, Tony. Can normal users look at all the problems? No. Because I showed that the CBio portal is pretty, I think, popular between biologists because they don't need to code and download the data. You can just. But at that website, there is no any option for you to color the plots based on the batches. If even they add one button, okay, color based on batches, you can see, oh, there's a batches. I don't want to go and validate that or something. There's a batch of X here. No, normal users can't see these things. Even getting the batch details was very, very difficult. Getting the times, how samples were processed, was very difficult to get those details as well.
Time for one last question. Okay. Check Slido, no more. All right, it just remains for us to thank Ramyar once again for a really entertaining and interesting talk.